Hi, everyone. You know enough about me now to probably talk about me to other people, so I'll skip the my name is Esha part and we'll dive right in. Um, that's okay. So essentially, my research is metabolic hormones influencing reproductive signaling. That is 5 words to simplify a very complex project, so I'll get started with the details. But before I do that, I'm going to bore you all a little about my personal life and acknowledge some of the people who made this happen. For one, all my friends who heard about my lab yaps and were asking why I was in the lab 24/7 every time they wanted to hang out. Thank you guys. Then comes my lab members. This is an old picture. There are more lab members, and not pictured is Dr. Chappelle, who you just heard from because he's pictured in person, as well as my advisor Wanda, who persuaded me into pursuing this second major. And then comes my family. They're very amazing people. All are pictured here, but especially my parents. They came to the country with nothing, built everything, and then especially my mom, who gave up her career so I could have mine. Now that I've put you all to tears, let's get started. So, the presentation roadmap. This is kind of how I'm organizing my entire presentation. It'll have this bar at the bottom so you can keep track of where we are because it gets a little messy. Let's start with the background information. Basically, in your body, your reproduction is a process that occurs. That includes puberty, pregnancy, lactation, sperm production, all of these things. They all require a ton of energy from your body. So that means like ATP. And in order for these things to happen, which they don't always have to happen because your body needs to be in an optimal state for them to occur. It's considered optional. If your body is not in an optimal state, these things will not occur. So you need high energy availability so it can support the activation of various reproductive circuits. Low energy means that you're going to trigger adaptive stress responses that's going to suppress reproduction and it'll try to conserve your bodily energy. So as you can see, we need to have our energy surplus be where it is so it can give adequate nutrients. It can keep your reproductive axis activated, which is what we want. Energy deficiency means you have limited nutrient availability and kind of want to stay away from that because then your reproductive axis gets suppressed. And your brain is like the central metabolic integration system for all of that. It is retrieving all these signals in the brain and then adapting them forward. So while I said that your brain needs or your body needs to be in an optimal energy state, that doesn't mean that a lot of energy is good energy. Or low energy is obviously bad. We know that. Deficiency in energy, like via stress, via lack of proper nutrition or chronic illness or any of these things, these are energy deficiency states in your body. However, there can also be energy excess, which means obesity, metabolic disease, and both of these things essentially suppress or disrupt reproductive signaling. So what we want to aim for is optimal metabolic balance and optimal energy balance so we can keep our reproductive state at its optimum. Okay. So now, moving on, to this part of the brain that I mentioned, it's the central integration of control. This particular region within the brain is called the hypothalamus of the brain. It's highlighted there in that image. And this is the control, the brain is obviously your control center, but the hypothalamus specifically is what connects the metabolic status to your reproductive output. So the hormonal signals, nutrient availability, the energy status in your body, and stress or inflammation or things that are going throughout your body all get integrated within the hypothalamus and it registers there like what's going on in my body. And then output is the reproductive output, which is things that we'll talk about like GnRH pulsatility, the hormonal release like LH, FSH, which is like the axis that I'll show you shortly, as well as your gonadal functions, so your ovaries and testes. The hypothalamus essentially determines whether metabolic conditions are favorable for reproduction or not. Now, here's the axis that I was talking about. So let me outline it as simple as I can for you. Starting at the top, we have our brain right up here. The hypothalamus is within the brain, as I just mentioned, and within these are KNDY neurons or candy neurons. They release GnRH pulses. So KNDY neurons, also kisspeptin neurons, I'll interchangeably refer to them. They release GnRH pulses, which is gonadotropin releasing hormone, and that acts upon the anterior pituitary gland. The anterior pituitary gland releases LH and FSH because of the stimulation it receives from the GnRH hormone. And then the gonads, the LH and FSH released by the anterior pituitary acts upon the gonads and like sex steroid production as well as gametogenesis, which is the production of gametes. So this is the axis we're working with today. Now, a little bit about GnRH, which is what I discussed in the previous slide. Essentially, it's really important for GnRH to be released in a pulsatile manner. It can't be continuous because if it's continuous, it will trigger a feedback loop that will make it so that LH and FSH is reduced and your fertility is impaired. That's not our goal. Our goal is to have normal fertility or optimal conditions for fertility. So you want to have normal LH and FSH and have this pulsatile GnRH release. So it's on point, on time. And that maintains your normal fertility. So the timing also matters, not just the amount. Now, going back to our candy neurons, they help us generate these reproductive hormonal pulses. So what they do is they coordinate pulsatile GnRH signaling. So we're focused on, I'll get to this in a second, but our study is kind of focused on more so the kispeptin and the neurokinin B that are released from KIS1 and TAC2 and 3 genes. Dynorphin is very important too, but that's not what my study looked at. Dynorphin is just inhibitory feedback, so I'll leave it at that. We don't need to focus there. Kispeptin is a compound produced from the KIS1 gene within the KNDY neurons. It stimulates the GnRH neurons that I talk about that act upon the anterior pituitary and then so forth, that axis, recalling that axis. And then we have the TAC2 and 3 genes that release neurokinin B, which helps synchronize the GnRH pulses, which the synchronization is very important, as we talked about. So all of these work together within the KNDY neuron to help this pulsatile GnRH release ongoing. Now, kispeptin, as I mentioned, is a major upstream regulator of the GnRH neurons. It's a key driver that activates these neurons and initiates these reproductive hormonal pulses. Kispeptin directly excites these GnRH neurons, and it's essential for their release of GnRH. It drives the pulsatile signaling, and it's very much required for fertility. So if you lose your KIS1 signaling, it impairs your reproductive function, essentially. Now, neurokinin B also helps coordinate that KNDY activity. It basically synchronizes the network and it continues the pulsing of the GnRH, and it's important for the rhythmic release, as I mentioned previously. So we need kind of both of them. So both of the genes that we look at in study, which I'll get to in a second, is KIS1 and TAC2 and 3. Keep that in mind. Now, metabolic hormones also act on, or probably may act on, candy neurons to alter fertility signaling. What this means is kind of transitioning into my study, which is the metabolic hormones of our body, which are already present in our body. Adiponectin and leptin are the particular focus of my study. There's a broad range of them, which we're also looking at in the lab, but I'll get to that in a minute. How do they act upon candy neurons specifically that then trigger the downstream pathways of our reproductive axis? We don't know the exact mechanism. So now going into what they exactly are, leptin is like signaling for your energy sufficiency in the body. So leptin means that energy stores are available. Lower leptin in your body indicates evidence of lower energy stores. Higher energy stores means there's an elevated amount of leptin in your body. So you want to make sure there's an adequate amount to be able to use that energy to then have this pathway ongoing. Now, adiponectin, lower means kind of like unhealthy, you're like insulin resistant, you have increased inflammation in your body, there's a lot of fat accumulation in that regard. And then higher adiponectin levels is kind of health, not like super high, but higher adiponectin normal levels are insulin sensitivity, which is good because your body can metabolize sugars, increase fat oxidation, as well as anti-inflammatory effects. So these are the two that we focused on for this study. Now, introducing kind of a third compound, but it's not a metabolic hormone, it's more of like a sex steroid, which is estradiol, or estrogen as we know commonly. It can also contribute to how neurons respond to these metabolic hormones. So within our bodies, we have a state of estrogen, and it's cyclical. So it's not always the same amount throughout the entire time. It's up and down especially for women and their cycles, it's very everywhere, actually, depending per person. So we wanted to know if this responds to metabolic hormones that we treat with as well, and how the hormonal environment of our bodies is contributing to the response. So it's an ovarian steroid hormone. It also regulates reproductive signaling, and it fluctuates across your reproductive state. Now, this is kind of leading into our research question, which is the direct effects of adipokines on candy-associated genes, because they remain unclear. So we know that adipokines, which are hormones made from fat, are the leptin and adiponectin, they influence reproduction in some sort of manner. And we also know that candy neurons regulate the GnRH pulses. What we don't know, and what we're kind of looked at in this study, is the direct regulation of the KIS1 gene, the direct regulation of the TAC2 and 3 gene, the interaction this has with the hormonal environment of your body, which is the amount of estrogen, or if it's present or not, as well as species-specific responses, because the two models we look at, which I'll introduce in a second, are murine and ovine models, which are mouse and sheep. So here's our research question. How do adipokines, leptin and adiponectin, impact KIS1 and TAC2 to 3 gene expression in murine and ovine KIS peptide neurons? Now this sets our entire stage for the hypothesis we have. So for leptin, as I mentioned, higher leptin means higher energy stores, lower leptin is lower energy stores in your body. So based on that information, it's kind of predicted that it would promote KIS1 and TAC2 through 2 to 3 gene expression in some sort of way. For adiponectin, it predicted to suppress KIS1 and TAC2 to 3 gene expression in some sort of way. Very general hypotheses. And then estrogen can modify these neuronal responsiveness. We thought that it would have some sort of effect, unsure what it would be. And because we're looking at two different models, the mouse and the sheep, we're expected to have some differences within the species themselves. So now moving on to the methodology, we use like an array of methodology. So I'll walk you through it. First comes the two models that we used, which are the KTAR1 murine mouse cells. This is what they look like under the microscope. These are immortalized murine candy neurons that come from the hypothalamus of the brain. So they look like that. And then for the ovine, OMBH is what the name is of the cell line, they look like that. We can notice that these are more like dense, and these are a lot more sparse, and that's just the nature of how they grow, how they are, and how they come out to be in a petri dish. But this allows us to do species-specific comparisons. Because we have only one of them, we can't really make those comparisons and see how they translate into different animals. Now, so the KTAR1 model, again, it's murine origin. There's kispeptin. It expresses the reproductive neuroendocrine genes. It's an immortalized model. This was done by the Chappelle lab, actually, in 2016, I believe. Done by a BRR student. And they immortalized this for us, so we thaw at our pleasure instead of going back to the rats and asking them for their brains again. So they have high proliferative capacity. They proliferate a lot more than OMB8 cells, so this means I can grow them like within a day or two if I want to, or I can let them. It's kind of risky to let them go for a week, otherwise they start dying on you. So you kind of like, you can do a lot of experiments at a lot faster rate with these cells. Compared to these ones, they take their sweet, sweet time. In about a week is when I can start experimenting with them if I wanted to. So we try to manage each of them accordingly. They both have different growth conditions and things like that. So maintaining the cells is a whole challenge and a separate project in itself. So they come from ovine, which is sheep, hypothalamic tissue. Both of them come from the same type of tissue in the brain, which makes it very cool to compare because we're seeing what happens sheep-wise and mouse-wise. Kispeptide, they have similar reproductive expression of the neuroendocrine genes I talked about. So KIS1 and TAC2 and 3 are both expressed in this model. They're also immortalized. There's no paper to refer to on this just yet. And they have a slow proliferative capacity. So they, like I said, grow a lot slower. Now, for the experimental treatment groups, we started with just adiponectin and leptin. This is what I got into when I first started, and I was like, we're done after this was over. We were not done. We had just started, actually. So these are what we had treated them with. They're recombinant, so we bought them off the store. online and treated it with various concentrations of adiponectin and leptin. And then in a separate cycle, or the same one, we also made dishes that were adiponectin and estradiol combinatory to see what that would look like if estrogen was present at that time. And we kept the estrogen concentration the same, so at 10 picomolar, so there's no variability there, just to see what would happen. All of these were treated for 24 hours in the incubator, and then the next day I came back to harvest them, and that brought us to our RNA isolation because the goal was to isolate DNA out of all of this. So the treated cells are right there. Then we harvested them the next day, came back to do RNA isolation, which is literally we isolated the RNA from all the cells that we had gathered. RNA in a second. Yeah, this is a long process. And then we did reverse transcription to then generate cDNA because cDNA is our goal. Now, cDNA can help us do a ton of experiments like these ones. So PCR. We used the cDNA to do PCR, and that we had a lot of primers we used as well, which is like the KISS1 primer and the TAC2 and 3 primers, because they bind to the cDNA and tell us, like, is it there or is it not? Or how much of the gene is there or how much of it is not? So I ran through the thermocycler, and then we were able to generate gel electrophoresis images, which you will see mine in just a second. The PCR products were separated on a gel, and then relatively, in the most perfect world, it would look like this. So you would be able to tell the difference between all the band intensities, and then no template control would basically be 0. Now, after that, that tells us if it's there or not and the intensity. What we did after that really tells us the quantification. So a qPCR was also ran, target like the same process we used, but in a different sort of format. So it would be able to output values for us, which are actual numerical values that tells us the differences between how much is being expressed under what treatment condition. And we always like quantitative data in science as opposed to qualitative, because it gives us more of an idea of like exactly how much differences there are. And so we were able to use that data, and using the delta-delta CT method, which is like a fancy way of saying we generated relative quantity values and assigned them to how much expression was done. And you will see all of this in a second, but I just thought to give you the process of the workflow before that. Now, for the results, there's a lot of layers to this, so stay with me here. First of all, we had to determine if KISS1 and TAC2 or 3 were even present. They should be, because they are present in the mouse and sheep brain as a whole. We just wanted to make sure they were present in our immortalized cell line. A lot of things happen in the process of immortalization. A lot of weird things happen. So wanted to make sure if they were being expressed in our cell line first. So up here, on this side, we have the KTAR. So this is a gel that shows mouse brain, very dark, which means we can see that it's present. And then in the KTAR one, which is our cell line, it's also present. And then these are no template controls. So they're a lot lower, and there should be ideally nothing on them because contamination. So good, there was no contamination in that. And then in terms of this one, OMBH, also we see that it's present in the sheep brain. The KISS1 and TAC3 genes are present in the OMB8 cells that we have, and it was clean. So this sets our stage for, now let's see what happens when we treat them with a lot of stuff. Now, in terms of treating them, we have the mouse adiponectin receptor and the mouse leptin receptor, as well as the sheep versions for both of those things. So before treating them, we wanted to make sure, are the receptors even present in our cells before we begin treating them with both of these things? If they were not, there's honestly like not a good valid reason for us to treat them with these things. So we found in that all of them were present as we wanted in a perfect world. So both the KTAR and OMBH, we saw that both types of receptors were detected. Now, we don't need to do qPCR for this because we just wanted to see if it was there or not. But if we wanted to know the quantitative value now, like we do in these ones, we would advance to qPCR. So here is the results for just adiponectin-treated KTAR cells, as well as OMB8 cells. You can see the representative gels above, and because I walked you through the gels previously, I'll trust that you know how to read them now and are experts at reading them. But here we were able to identify that, oh, it's showing up. But here's where our interest lies in, like, how much did it really show up? So as you can see, like, this is no adiponectin, which is our control, and increasing the doses of adiponectin gives us a differential gene expression of the KISS gene. And similarly, in the ovine, we can see the differences as well, depending on the concentration of treatment that we gave the cells. Now, it's not all the same, and obviously we did not expect that either, but it gives us an idea of like the general trend. So what I'm seeing based off of the zero treatment is that adiponectin is kind of generally doing a suppression of the KISS1 and TAC3 genes, 2 to 3. Now, MTAC2 was a little bit difficult for us because it had a lot of non-specific amplification as well as weird curves that were showing up. And after many tries of qPCR, I've decided that it was not going to be included in this and it would need continued further optimization. So moving on, estrogen modulated it as well. So we can see that now that we've added estrogen into the mix, there are a lot of different expressions for both the KISS gene as well as the KISS and TAC3 gene. So there are different than what we saw right here. So we know that estrogen is having some sort of modulatory effect when we're putting it into the mix. Now moving on to leptin. Leptin, we also saw a huge difference depending on the concentration that we treated it with. So above are the representative gels, and the gels correspond here and here and here. We see that compared to the zero treated, leptin generally has an expressionary effect, so it expresses higher than the zero treatment control. But again, these are just general statements, but we can see within each concentration, it's very different expression of both the KISS1 and the TAC3/2 genes. Now for the estrogen, we also did the same thing as we did for the adiponectin. We treated it again, and then we use the same cDNA to generate these graphs. And we can still see like it's not the exact same as it was previously. So that means that estrogen has some sort of modulatory effect as well. And these are the representative gels for that above. So that was a lot. I threw a ton of information, and you probably don't even remember it. But the big idea is that the adipokines that I talked about, which are adiponectin and leptin, they alter KNDY-associated gene expression in the murine and ovine neuronal models. So leptin, we saw generally had an increased reproductive associated gene effect. So it was generally increasing compared to the zero treatment that we did for adiponectin. Both for KISS1 and TAC2 in both models. This is a very general statement. Similarly, for adiponectin, it decreased reproductive associated gene expression, so both the KISS1 and TAC2/3 genes were a lot less than they were in the zero adiponectin treatment, and it suggests that there was modulation during negative energy states. As for estradiol, we saw that there were altered compared to when it was no estradiol, which means that estrogen may be having some sort of modulatory effect within there as well. Now, murine and ovine, they are two different various species, as I mentioned. They also responded very differently. In an ideal world, they would respond the exact same, and then we can move on to clinical trials, but that did not happen. So they did respond very differently, which gives us insight because both models are very different. They were also immortalized in different years, different conditions, and everything. So keeping that in mind. So basically, all in all, these findings support that direct metabolic regulation of reproductive neuroendocrine signaling through KNDY associated pathways. So connecting that back all to what I showed you earlier, metabolic hormones integrate into the HPG axis. So basically, adiponectin and leptin have an impact on these KNDY neurons, as we have seen through our graphs. We see that it's generally suppressatory with adiponectin, generally expressatory with leptin. It kind of stimulates or suppresses the neuronal activity and gene expression. And because it does that all the way up there in the KISS peptin area, that means the rest of our pathway is going to respond accordingly. So if our KISS1 gene expression or TAC2 or 3 gene expression is increased or decreased, then it's going to affect the anterior pituitary, or first of all, it's going to affect the GnRH neurons, and then the GnRH neurons are then going to affect the anterior pituitary, and that's going to affect the release of LH and FSH, and that's going to affect our gonad. So we start all the way up here so we can see the downstream effects of how it could possibly impact our entire reproductive axis. Now, so from mechanism to medicine, which is like a simple way of saying how do these very small scale biochemical aspects impact us clinically, which is probably what you guys all care about anyways. So we know that metabolic dysfunction or disrupted hypothalamic signaling or reproductive dysfunction, all of these things are prevalent, whether we, depending on our diet, depending on our lifestyles or exercise, we all have particular metabolic states. We could have disrupted hypothalamic reproductive signaling and not even know it. And that could be impacting our reproductive dysfunction. So a lot of conditions such as obesity, diabetes, PMOS, all these things, they are all related to our metabolic state within our bodies. Now, we could probably use not my data because this is very preliminary, but down the line when this is replicated and more verified, we would use this information to possibly understand what kind of hormonal state in our body is optimal for reproduction. So leptin and adiponectin are just two we looked at for the purpose of the thesis, but there's very many more like insulin resistant and IGF, and how the combinatory effects of all these hormones can impact our candy neurons and how that impacts our GnRH pulses that we first talked about when we first started. Now that could help us understanding what the optimal condition is, can help us develop therapeutic approaches. So metabolic modulators, we can use these to help optimize the reproductive state, hypothalamic targets, and then eventually we could restore reproduction or help our health in some sort of way, more generally keeping it a little lower. It could possibly unlock new therapies for reproductive disorders. Not saying we can cure anything right at this moment, but we can try. Now, as all good studies do, we did have some limitations. So these were in vitro models, as I mentioned. I didn't move outside of a petri dish when I was doing all of these things to all of these cells. So it is very contained to just neurons. It is not a broader within like an animal. We didn't see any interactions with the rest of the brain or the rest of the body. So we have to keep that in mind that they were isolated cultures, so we couldn't really measure anything downstream of what I talked about. We stopped at leptin neurons. There were measurement limitations as well. So we looked at RNA, cDNA. We didn't really look at like protein or anything down the line, which is kind of important to do, obviously. And our neuronal activity was unknown as well, because that's not the scope of this lab or my study. So our experimental scale was also pretty limited. We only had one biological replicate for everything that we did in the study, though many are being generated at this time as we speak. Not right now because I'm not up there, but there were some primer optimization challenges as well. As I mentioned with the MTAC2, not everything goes perfectly. You have to order multiple primers to be able to find the perfect one within even the same gene. So we're continuing to optimize those. And then the ovine cultures obviously required a lot of care, more like time as well. So that's why the slower generation of those neurons to health and therefore treatment took a long time. Now, translational limitations, obviously there were the species differences that we saw. We can't like say that these results are particular for everyone. And we don't really know the long-term outcomes of anything. This is a very preliminary study, as Dr. Schell mentioned in the very beginning. But they highlight the need for future studies to validate and build upon these current findings, which we're doing right now and will probably do even after I leave. And the future directions are such. So we're looking at a lot of the other things I mentioned, like insulin, IGF, resistin, GLP, how they affect and therefore their impact on this gene expression of not just KISS and TAC, but also the other receptor genes that are present in the KNDy neurons. Can move beyond mRNA, probably not this lab, but other labs can take our data and do that as well as do electrophysiological studies to understand, because neurons fire with each other and among other things, and that's a whole different scope of study, like neuronal electrophysiology. Other labs can build upon with that. Temporal context, we are all on a circadian cycle at all times, knowing when treatment or how long treatment, because I only did 24 hours. It could be that 6-hour treatments or 12-hour treatments have different effects, so understanding that, and as well as like a translational impact, which is for later. But these are all things that still need to be done. Now, tying everything together, now you guys know so much about reproductive neuroendocrinology, but basically the main message and the take-home message is that the HPG axis, the hypothalamic pituitary gonadal axis, hypothalamic, it integrates the metabolic, neuroendocrine, and reproductive signals all together to help determine reproductive health and all these things. So it's really important to study that area of the brain specifically, as well as understanding all these connections. It helps pave ways for personalized therapies and healthier futures way, way down the line. Um, thank you. Any questions?